Hello everyone, I am Dr. Amer, MD in Pathology, and I am an Assistant Professor of Pathology. Welcome to today's lecture on Neuro Embryology. Neuro Embryology is one of the most important topics in embryology because it explains how the brain and spinal cord develop and helps us understand many congenital abnormalities seen in clinical practice. In this lecture, we will learn how the nervous system develops from a simple layer of embryonic cells into the complex human brain and spinal cord. I repeat. We will learn how the nervous system develops from a simple layer of embryonic cells into the complex human brain and spinal cord. This concept is the foundation for understanding neuroanatomy, neuroscience, and many neurological disorders. Throughout this lecture, we will cover formation of the neural plate, neurulation, neural tube formation, neural crest cells, development of the brain and spinal cord, brain vesicles, ventricular system, major congenital anomalies with clinical correlations. Again, remember this important point. Neuroembryology is the study of how the nervous system develops from the embryo into the mature brain and spinal cord. Let's begin our fascinating journey into the development of the human nervous system. Now, before we study each stage in detail, let's first look at the overall timeline of nervous system development. This slide gives you the complete roadmap of neuroembryology. I want you to remember one important fact. The nervous system is the first organ system to begin developing, but one of the last to complete its maturation. Let me repeat that. The nervous system is the first organ system to begin developing, but one of the last to complete its maturation. Now let's follow this timeline. On day 18, the neural plate appears. This is the very first sign that the nervous system is beginning to develop. Around day 20, the neural plate bends inward to form the neural groove, while the edges rise to form the neural folds. By day 22, these neural folds start to fuse, forming the neural tube, which will later develop into the brain and spinal cord. This fusion does not happen all at once. It begins near the cervical region and then continues in both the cranial and caudal directions. Around day 25, the cranial or rostral neuropore closes. If this closure fails, it results in anencephaly, a severe congenital malformation. By day 27, the caudal neuropore closes. Failure of this closure leads to spina bifida. Moving into week 4, the developing brain enlarges and forms the 3 primary brain vesicles. Then, during week 5, these further develop into the 5 secondary brain vesicles, which eventually give rise to all the major parts of the adult brain. Again, remember these milestones. Day 18, neural plate. Day 20, neural groove. Day 22, neural tube begins forming. Day 25, rostral neuropore closes. Day 27, caudal neuropore closes. Week 4, primary brain vesicles. Week 5, secondary brain vesicles. Don't worry about memorizing everything right now. We will study each of these stages individually, step by step, in the next slides. Now that we have seen the overall timeline, let's begin with the first stage of neuroembryology. Everything starts during week 3 of embryonic development with a process called gastrulation. Gastrulation is one of the most important events in embryology. Let me repeat that. Gastrulation is one of the most important events in embryology because it establishes the 3 primary germ layers that give rise to every tissue and organ in the human body. Initially, the embryo consists of only 2 layers, known as the bilaminar embryonic disc. During gastrulation, a 3rd layer forms, converting it into the trilaminar embryonic disc. These 3 germ layers are ectoderm, the outer layer, mesoderm, the middle layer, endoderm, the inner layer. Remember this point because it is asked frequently in examinations. The entire nervous system develops from the ectoderm. I repeat, the brain, spinal cord, and peripheral nervous system all originate from the ectoderm. Now let's move to step 2, notochord formation. The notochord develops from the mesoderm and acts as the primary organizer of embryonic development. Although the notochord does not become part of the nervous system, it is absolutely essential for nervous system formation because it sends molecular signals to the overlying ectoderm. The most important signaling molecule released by the notochord is sonic hedgehog, commonly abbreviated as SHH. This signal instructs the overlying ectoderm to transform into neuroectoderm, which is the first committed nervous tissue. Without the notochord and its signaling molecules, the nervous system cannot develop normally. So remember these 2 high-yield concepts. Gastrulation forms the 3 germ layers. The notochord induces the ectoderm to become neuroectoderm through sonic hedgehog SHH signaling. In the next slide, we will see how this neuroectoderm thickens to form the neural plate, marking the beginning of neurulation. Now that the ectoderm has been induced by the notochord, the next stage of development begins. The ectoderm directly above the notochord starts to thicken and forms the neural plate. Remember this important point. The neural plate is the first visible sign of nervous system development. Let me repeat that. The neural plate is the first visible structure that marks the beginning of the developing nervous system. As development continues, the center of the neural plate sinks inward, creating the neural groove, while the edges rise upward to form the neural folds. Think of it like folding a flat sheet of paper into a tube. As these neural folds continue to move toward each other, they eventually fuse in the midline to form the neural tube. This process is called neurulation. The neural tube is extremely important because it gives rise to the entire central nervous system, including the brain and spinal cord. I want you to remember this. The neural tube forms the brain and spinal cord. After the neural tube forms, both ends remain temporarily open. These openings are called the neuropores. There are two neuropores, the anterior or rostral neuropore, which closes around day 25, the posterior or caudal neuropore, which closes around day 27. These closure times are very important for examinations. Now let's discuss the clinical significance. If the rostral neuropore fails to close, the result is an encephaly where much of the brain and skull fail to develop. Unfortunately, this condition is incompatible with life. If the caudal neuropore fails to close, the result is spina bifida, a neural tube defect that affects the spinal cord and vertebral column. The severity can range from mild forms, such as spina bifida occulta, to more severe forms, such as meningomyelocele. Fortunately, many neural tube defects can be prevented. Adequate folic acid supplementation before conception and during early pregnancy significantly reduces the risk of these defects. So, before moving to the next slide, remember these 5 key points. Neural plate is the first visible sign of nervous system development. Neural groove forms by invagination of the neural plate. Neural folds fuse to form the neural tube. The neural tube develops into the brain and spinal cord. Folic acid helps prevent neural tube defects. In the next slide, we will study neural crest cells, one of the most fascinating cell populations in embryology, and discover the many structures they form. So far, we have seen how the neural tube forms the central nervous system. But an important question remains. Where does the peripheral nervous system come from? The answer is the neural crest cells. Neural crest cells are a unique group of cells that arise from the edges of the neural folds during neurulation. After the neural tube closes, these cells separate from the neural tube and begin to migrate throughout the developing embryo. Remember this important point. Neural crest cells are highly migratory cells that form structures throughout the body. Let me repeat that. Neural crest cells are highly migratory cells, and because they travel to many different parts of the embryo, they give rise to a wide variety of tissues and organs. Now let's look at their major derivatives. First, they form most of the peripheral nervous system, including the sensory ganglia, autonomic ganglia, Schwann cells, and satellite cells. Second, they form melanocytes, the pigment-producing cells responsible for skin and hair color. Third, they contribute to the adrenal medulla, which produces the hormones epinephrine and norepinephrine. They also form important craniofacial structures, including parts of the facial bones, cartilage, connective tissue, and the odontoblasts that produce dentin in teeth. In addition, neural crest cells contribute to the septum of the outflow tract of the heart, making them essential for normal cardiac development. Now let's compare the neural tube and the neural crest. The neural tube forms the central nervous system. That means it develops into the brain and spinal cord. The neural crest forms most of the peripheral nervous system and several other important structures throughout the body. I want you to remember this comparison because it is asked frequently in examinations. Neural tube equals central nervous system. Neural crest equals peripheral nervous system and many other tissues. Finally, remember that defects in neural crest cell migration can lead to important clinical disorders such as Hirschsprung disease, DiGeorge syndrome, Waardenburg syndrome, and neuroblastoma. These conditions highlight how essential neural crest cells are for normal embryonic development. In the next slide, we will begin exploring how the developing brain is organized into primary and secondary brain vesicles, which eventually give rise to all the major regions of the adult brain. Now that the neural tube has formed, the next question is, how does a simple neural tube become the highly complex human brain? The answer lies in the formation of the brain vesicles. Remember this important point. The brain develops from the cranial end of the neural tube. Let me repeat that. The brain develops from the cranial end of the neural tube, while the remaining caudal part develops into the spinal cord. During week 4, the cranial end of the neural tube enlarges and forms 3 primary brain vesicles. These are prosencephalon, also called the forebrain, mesencephalon, or the midbrain, rhombencephalon, also known as the hindbrain. These are called the primary brain vesicles because they are the first divisions of the developing brain. However, development does not stop here. During week 5, these 3 vesicles further divide to form 5 secondary brain vesicles. Now let's understand each one. The prosencephalon, or forebrain, divides into 2 parts. The first is the telencephalon, which later develops into the cerebral hemispheres, including the cerebral cortex, basal ganglia, and lateral ventricles. The second is the diencephalon, which forms the thalamus, hypothalamus, epithalamus, retina, optic nerve, and the third ventricle. Notice that the mesencephalon does not divide. It remains as the midbrain and contains the cerebral aqueduct, which connects the third and fourth ventricles. The rhombencephalon, or hindbrain, divides into 2 parts. The metencephalon forms the pons and cerebellum together with the upper part of the fourth ventricle. The myelencephalon develops into the medulla oblongata and the lower part of the fourth ventricle. Everything below the myelencephalon continues as the spinal cord, containing the central canal. Now let's summarize this in a simple way. 3 primary brain vesicles become 5 secondary brain vesicles. I repeat, 3 primary brain vesicles become 5 secondary brain vesicles. The forebrain becomes the telencephalon and diencephalon. The midbrain remains the mesencephalon. The hindbrain becomes the metencephalon and myelencephalon. If you remember this sequence, you can easily understand the origin of every major part of the adult brain. In the next slide, we will examine these brain regions individually and understand how each one contributes to the structure and function of the mature nervous system.